So we have a 2-for-1 in this next presentation. Both Ann Carpenter and Shantanu Singh are going to be giving a presentation. They co-lead a lab here at the Broad focused on learning using biological images for drug discovery. Ann co-invented the open source cell profiler software used by thousands of biologists worldwide and cell painting assay that is now widespread in the academia, in both academia and industry. Shantanu Singh has been leading the development of machine learning methods for image-based profiling and cell painting. They lead the JUMP Cell Painting Consortium, which created the world's largest public cell painting database to help accelerate the search for promising drug candidates. The title of their talk is Finding Disease Phenotypes and Candidate Therapeutics Using Images Cell Painting.
Thanks.
So, Ann?
Hi, everyone. Oh, is this not displaying the slide? Okay. All right, I'll look back at it then. Thanks.
You're just going to have to remember what your slides are.
I guess so. Thanks, everyone. I'm really happy to be here today to tell you about the work that's going on in the Carpenter Singh lab here at the Broad Institute, and we focus on images. And I apologize in advance to those of you who follow our work routinely, because today's talk will be quite an overview, big picture presentation, because I want to make sure that the concepts that we're working on are become familiar to you by the end of the talk. So Shantanu will talk about some of the newer stuff towards the end, but I want to begin with giving you a sense of context. Also, as well, you can download a PDF of these slides so you don't have to furiously write down citations, but you're also welcome to take screenshots or anything that you would like during this presentation. So just for context, our lab works in the space of large-scale imaging experiments. And these are experiments where we're doing different perturbations in each well. It might be a different patient cell line in each well, but more commonly, it's something like a compound or a genetic perturbation, CRISPR or overexpression of genes within each well. And each well gets thousands of cells. They're imaged using automated microscopes that are quite reliable these days. And so it's a pretty robust kind of experiment to do. And then we use image analysis software to sometimes extract particular features. Sometimes we use deep learning to extract feature embeddings that capture the information in the images and are maybe a little less interpretable. But either way, the goal is to capture that information content about what's the cell's status, what pathways are active in the cell, what's going on within the cell as measured by the morphology that it takes. And morphology here we're defining quite broadly to include things like the size and shape of different organelles and the cell itself, but we're also capturing things like the intensities and textures of different stainings and their relationships to each other. So that's a basic premise. And if you're familiar with images being used, of this high throughput imaging being used in drug discovery, it's probably going to be in the context of high content screening. And that term refers to the idea of using a microscopy assay as a readout for some particular biology that you care about. And this field has really matured over the past 20 years or so, to the point where it's now quite routine to do this in mammalian cell types. There's really challenging edge cases, like some neuronal cell types are really hard to trace. There's fancier and fancier physiologically relevant models systems like 3D and time-lapse that can be really challenging to quantify. And this research is where I started my career and now is being taken on by a former alum of the group. That's Dr. Beth Simony. And her lab now focuses on some of the things that we've started together. So this includes Cell Profiler, which is this open source software. The new generation Piximi is based on deep learning, which allows classification and eventually will allow segmentation as well. And this work is happening because of this lovely NIH center that funds this work. And so I won't be talking about this kind of image analysis to measure particular features that biologists want to measure. I'm instead going to be moving in a different direction, but I do want to say that this kind of work has had clinical impact already. Certainly many pharma companies have used high content screening. Many of them have used our software to identify particular phenotypes they care about in images and then found hit compounds that they've taken from there. I just want to focus on two particular examples, one that we were involved with. The first one here is a successful clinical trial for myelofibrosis. It was a repurposing experiment where we helped these researchers identify compounds that could make These out-of-control cancer cells become polyploid, and you can see the obvious visual difference between the top and the bottom there. The second example we had absolutely nothing to do with, except they used our software in the trial, and it was very nice. Andrew Hopkins this morning focused on this at the beginning of his keynote. This was the personalized cancer medicine project that he talked about, made use of our software. So we're really proud of the impact that that has had on the world. And as I said, I'm going to shift now. Instead of helping biologists measure phenotypes they know that they care about already, we're going to talk about using images in a more machine learning kind of way, where we profile the sample to characterize many features of that sample, and then you compare the patterns that we see in the data to accomplish various tasks in drug discovery. And there's all kinds of different applications. We'll just touch on a few of them, but we've left a lot of time for questions, so we're happy to elaborate on anything. So the basic concept of using images as a profile, as opposed to just measuring particular features of interest, is maybe easier to understand than the context of thinking about how diseases are diagnosed. So many of us can tell a child that has Down syndrome based on the appearance, the morphological appearance of the child's face. And sometimes you have to be quite an expert to be able to discern different diagnoses from a face, such as Engelmann, for example. But this is a task that machine learning can do very well at for many different disorders, just looking at the human face. Sometimes you can't look at a person's face and diagnose their disease. You actually need to dig a little deeper, and that might be zooming in, it might be taking blood samples, it might be looking more closely at their cell structures, but the fundamental concept is the same. There's something about the structure or morphology that is telling you what's going wrong with this disorder, and if we can do this if we can make that kind of a diagnosis in a dish with cells growing away from the patient, we have a potential to screen drugs in order to potentially correct that disorder. So that's a basic concept. In image-based profiling, we use these images to create signatures of genes and compounds and diseases. So this is the most important slide, just to get the concept of what we're doing here. We have many perturbations, usually hundreds of thousands. And as I said, they can be patient cell lines or compounds or genetic perturbations. We carry out some kind of imaging assay. We often use cell painting, and I'll talk about that in a second. And then we extract features. Again, they can be classical features, or they can be deep learning-based embeddings that allow us to capture the information content of the image. So what do these cells look like? It's single cell level data. You can see this is real teeny tiny snapshots of real data from a real experiment. You can see that instantly there's a difference between sample 1, 2, and 3 in terms of the patterns of the features that are extracted, just like we would see for mRNA or proteomic profiling. And once we have those profiles extracted, we can then look at the similarities and differences between different samples, and we can, as I said, do all kinds of things with that sort of data. So this is the basic concept of image-based profiling, very similar to mRNA profiling or proteomic profiling in terms of the kinds of applications and also the kinds of challenges in carrying out these experiments. The actual image assay that we often use is called cell painting, and it was designed with one purpose in mind, which is to make a very inexpensive, very non-fussy assay that any biologist could carry out in pretty much any laboratory, image on a fairly straightforward microscope. You just have to have the right filter set, and very, very, very high throughput. So it's not impossible for a pharma company to screen 5 million compounds in their deck through this one assay. It's not expensive, and it's not difficult to carry out. The assay was designed with just dyes. There's no antibodies, so there's not wash steps and so on. It's really straightforward from that perspective. And it captures the basic organelles. So these are the kind of a cell biologist's favorite set of organelles in order to look at the basic structures. And when we extract data from all these different channels within the images, we can use these profiles to link drugs and genes and diseases. All right, so throughout the rest of the talk, we'll just go through some different applications and examples, and again, happy to elaborate on any of them that interest you. The first one is, can we identify signatures of disease? And if you can identify a signature of a disease in a dish with cells that are growing away from a patient, then you can certainly screen drugs, test them one by one to figure out which drugs might be useful to reverse that signature. So let's look at an example of this. Here, we worked in collaboration with Mikko Taipale at the University of Toronto. Before we met, his graduate student, Jessica Lacoste, had been working very hard by eye, detecting the difference between these two states, and I'll describe them now. So She would take a protein that had been associated with a human disorder, where a mutation in that gene was associated and known to be causative for a human disorder. She'd take the wild type version, the normal reference version of that gene, and overexpress it in cells, and it has a potential to tag it with a fluorescent label. And so this is what this distribution of this one particular protein, LZTR1, looks like. And then she would make the same exact protein, but make the one mutation that's known to cause a human disorder, in this case, Noonan syndrome. And you can see if there's basically three black oval holes here that are the nuclei, and whatever this protein is doing, it's located in these funny blob shapes a little outside the nucleus. But you can see when the mutation is present, you still see the nucleus in the middle, or the absence of staining in the middle. So the protein's still in the cytoplasm, but now there's this kind of diffuse staining, and the blobs definitely look very different. So in this experiment, again, you could also, in this particular case, diagnose this disorder by looking at a human face. Here, we're just looking at cells that have been engineered to, or in this case, transiently transfected, to express either the regular protein or the mutant protein. And so what's lovely about this kind of an experimental setup is that you do not need to be an expert on this syndrome. And that'll be a theme throughout today's talk, is that we're looking at really systematic approaches across lots of diseases at once. We don't need a lot of customization. In this one experiment, you can just say, well, I may have no idea what this protein's normal function is. I may have no idea what the symptoms of this disorder are. But I can still, nevertheless, see there's a very obvious visual difference between these two. So she scored this experiment by eye. And it actually involved thousands of different reference proteins and then their mutant counterparts. The nice thing about this kind of a setup is that maybe you don't see a difference between the two all the time, but for those cases where you do, you can then test drugs to try to reverse the phenotype. And so just to give you a sense of the status of the project right now, she's tagged and overexpressed and imaged thousands of these disease variants across 1,000 different disorders. And as I said, it's not all the time that you see a change. By collaborating with ourselves, including Marzia Hagigi, a postdoc in the lab, she's been able to quantitatively identify changes in not just the localization of the protein that I show here, but a couple other channels of the image that show other parts of the cell. And so far has discovered a couple hundred disease phenotypes. So as a fraction of the whole, it's a small fraction, less than 10%, but as an absolute number, it's huge. So to say to researchers that are studying these diseases, hey, we found a phenotype that is induced when this causative mutation is present. We have a morphological phenotype. It's useful for two reasons. Number one, maybe if you actually know what are these structures and what is happening, is it a trafficking problem? Is it an expression problem? Does it impact the proteins, the amount of the protein present? That kind of mechanistic information can be very useful for those studying the diseases. But second, as I said, you can now do a drug screen. So here just gives you a picture of some different examples. It's very systematic to do this across thousands of disease variants all at once. And what's even more exciting / terrifying is that in the next 4 years or so, we'll be generating this kind of data for 80,000 human genetic variants, some of which are known to cause human disorders, others of which have an unknown impact. And so this project will be yielding not just the mechanistic information and not just the potential for drug screening, but also information about whether this variant is likely to be causative or whether there are a given disorder might have two sets of variants that do different things to the function of the protein. So those are the kinds of things we'll be able to discern here. And just want to comment on this project that it creates a new bottleneck. So when we have when I say, okay, now we have a couple hundred disease phenotypes, we'd like to do a couple hundred drug screens. And so it's a really neat collaboration that I won't show today, is collaborating with JT Neal and Paul Blaney here, who have a neat barcoding-based optical profiling method that allows us to take cells like this, mix 200 of these different genetic variants together in one well of a dish, treat with a compound, and then just say, does does this compound seem to reverse the unhealthy phenotype for any of these 200 disorders at once? So that pooled kind of screening is the next step that we're hoping to take to overcome this bottleneck that we're making. We have 200 phenotypes now, but once we analyze these 80,000, we'll have on the order of 4,000 or 5,000 to analyze in that kind of a way. So I described there are many ways to identify disease phenotypes using image-based profiling. I described this one here using variance localization. We've also done an experiment using patient cell lines for patients who have different mental conditions and identifying the morphological changes that are associated with psychosis, and really excited to turn that to the next step of doing drug screening. And this overall principle has been proved by Recursion, which is a company that is based on some of the technology coming out of our lab. And I serve on their scientific advisory board. They take a different approach, which is underexpressing genes, so knocking out genes originally with RNAi, but then later with CRISPR, in order to identify, again, in a very systematic, very high throughput kind of way, what are the morphological phenotypes that are associated with different diseases, and then turning those into drug screens. So lots of ways to do it, but the same basic concept is let's find screenable disease phenotypes in a dish that we can test compounds on. So the next example I'll turn to is identifying gene and allele functions using cell painting. And I guess I should pause here for a moment. It might be a little confusing because I talked about cell painting as a particular set of dyes, but then I talked about, now we're localizing with a particular stain. So a lot of what I'm going to talk about today, it really doesn't matter what the stain set is that's in the experiment. So the actual particulars of the assay are not super important. I really just want to convey the concept of using whatever images you have, whatever assays you have. It doesn't have to be cells. It can be zebrafish movies. It can be whole body scans of mice. Whatever imagery you have, it's possible to extract all those morphological features from those images or movies, and then take this basic approach of which samples look like each other and which ones look different from each other to accomplish different goals. So another goal we might want to accomplish is to figure out a gene's function and then ultimately a lot of different allele functions by grouping them together. So if you have a query gene or an allele, you've extracted its features, can we match that against a library of known, a database of known perturbations and figure out which ones match with each other? So our original experiment for this was finished 5 years ago, where we overexpressed a very small set of genes and found that roughly half of the genes tested. It was a somewhat cherry-picked set of genes to be more well studied than the rest of the genome. I don't expect 50% to be true for the whole genome, but it was pretty shocking to us at the time to find such a large proportion of genes in this one assay at this one time point in this one cell type. This roughly 50% of the genes were distinguishable from negative controls based on their morphological impact. So in this first experiment, we were really excited to see that rate, and even more exciting to see that those, it wasn't like all the genes looked like each other. They fell into really nice clusters, and those clusters had really strong biological meaning. So for example, the hippo pathway is over here, and we found even negative correlations, which it was quite surprising to a lot of the biologists and others on the team that even negative correlations made biological sense. So if you had a positive and a negative regulator for the same pathway, they would give opposite morphological phenotypes. And by opposite, I mean kind of anti-correlated features. So this was laying the groundwork for what is a pretty exciting direction, which is called, it's a form of virtual screening. There's a lot of ways to do virtual screening. We've heard some of them today. And in this case, we're using just image-based data to do screens, and we're doing it based on a gene name, a gene that's of interest. So what we can do, if you have a gene where you knock it out or you overexpress it and you get a really distinctive phenotype, there's nothing stopping you from going and looking at a gigantic compound database of cell painting data and seeing if you can find a match, exactly analogous to transcriptional profiling using, let's say, the connectivity map data. And this has been really exciting to give a try. We weren't sure it was going to work, but we're really excited to see that it does, where we did it in, again, a very small pilot experiment where we had had only 69 genes that had met this criterion of having a nice cell painting phenotype. And we tried to match them against a pretty small set of compounds, around 30,000 compounds, roughly 15,000 of which were distinguishable from negative controls. So this is the raw material, the data that we were using for this matching. And we were looking for matches and also anti-matches. We were looking for correlations and compounds that highly correlated with the gene overexpression, and also compounds that highly anti-correlated with the overexpression. And so in the project, we ended up testing, first of all, of course, on known gene compound pairs where it was already known that this compound should target the activity of this particular gene. So we had 63 of those positive controls built into this experiment. We found roughly 32% of them had the correct compounds at the top of the list. And by top, I mean top and bottom, because it's correlating and anti-correlating. And so then that's all well and good, but as has been mentioned, the proof is when you actually try to do a prospective test. And so in a prospective test, using genes where we did not know what the answer would be, we grabbed 7 genes, and we made lists of compounds, and then we ran around, not just the Broad, but worldwide, contacted a few laboratories that were studying these genes and said, hey, we have this method, and it looks like we can pluck good compounds, 30% of the time at the top of the list, one of these compounds is likely to impinge on this pathway that you study. Would you like to test these compounds? And so we found 7 labs that were willing to give it a try. 5 of them gave good results. 2 of those fell apart for chemistry, so I haven't detailed them here. But 3 of them just really worked. The top of the list had some compounds that impinged upon the pathway that the researchers were studying. That was the expected pathway. One of which was especially exciting, a collaboration together with Karen Eisinger at UPenn focused on these Hippo pathway regulators, YAP1 regulators, where we found 3 really nice regulators, and then we could do a teeny bit of medicinal chemistry using some analogs that already existed in our library, testing those and finding that we could kill sarcoma cells. So these are a YAP-dependent sarcoma cell line that's really hard to kill. This is a type of cancer that's really hard to treat, and she's been really thrilled to find compounds that have, in these blue were our original hits, and then we found some even stronger ones that seem to impact this pathway. We don't know if they bind YAP1 directly, but impact this pathway and reduce the growth of this sarcoma cell line. So we're excited to follow those up. The next project that we'll talk about is assessing the mechanism of potential drugs based on their profiles. And here's where Shantanu will take over.
Thanks, Anne. I believe I have it all set. Okay. So now learning about a compound's mechanism of action by just matching the image-based profiles to a known library of compounds that has sort of been demonstrated almost a decade ago if you've been following this literature. And it's used in many pharma companies, but what we are curious about is how would this compare to doing the same thing, the same idea, just matching using mRNA profiles, which are more expensive, but also capture a different layer of cell state. So in this recent paper, we compared 2 different high-throughput profiling assays, cell painting, which captures morphology, and L1000, which captures the transcriptional state of about 1,000 genes in high throughput. We profiled about 1,500 compounds, mostly FDA-approved drugs from the Broad Drug Repurposing Library across doses, and we found that across these 2 assays, compounds in about 1/3 of the mechanisms that we tested were sort of significantly tightly clustered. So in this analysis, we found that morphology was consistently higher than L1000 in terms of the number of mechanisms it captured. You can see the orange being taller than the light blue in the plot, with the dark blue being the overlap. So that's when you sort of do an unsupervised analysis. You just sort of rely on the matching that comes out of the representations that you have. And you measure the similarities and you report those metrics. The story is a little bit more balanced when you do a supervised analysis, where you train classifiers for this task of prediction. The 2 axes here, it's blurred, represent the some classification metric, the area under the precision recall curve for these 2 assays. So we find, not surprisingly, that some mechanisms are better captured than others. So there's complementarity between them. This work is our most thorough investigation to date on comparing these 2 assays in general, not just on the task of mechanism of action prediction, so you can look up the paper for more details on the rest of that. Now, cell painting can also be used for predicting other assays. Large-scale assays for drug discovery can be expensive and impractical, particularly those that involve model systems that require primary cells, organoids, or whole organisms. But as an alternative, an international team, including Janssen and many others, a few years ago had demonstrated that data from a single high-throughput imaging assay can be repurposed to predict the biological activity of compounds in other assays, even those that are completely unrelated to the biological process that was being studied in that initial imaging assay. So we've extended this in our own work, where we combine imaging with chemical structure and transcriptional readouts to predict these assay readouts. We found that fusing multiple data sources didn't work so well, so we instead simply individually picked what sources work best for any given assay. So this proof of concept result here suggests that image data from high-content cellular assays, when combined with other available information, can be leveraged to carefully design compound screens and boost hit rates. And I'd like to acknowledge Regina's group for having contributed to formulating this problem with us, and Kevin Young from her group was participating in this project. Cell painting can also be used to predict markers of cell health. Paquita Vasquez and others at the Broad had created a cell health assay that reads out a set of 70 or so phenotypes, including the proportion of cells in a specific cell cycle stage, the kind of apoptosis, initiated cell proliferation rates, reactive oxygen species, and many other relevant readouts of cell health. We collected genetic perturbation data for 3 cell lines for a small handful of genes in both the cell painting and the cell health assay. And then we use the cell painting features to predict the cell health phenotypes. So we can do a really good job of predicting several of these readouts, and you can learn more about the individual readouts as well as the source code, as Pat would want us all to have online. The assay prediction application that I just showed, as well as the virtual screening that Anne had referred to, are both sort of data-hungry applications, right? The more data you have, the likelihood of your performing better improves. So in this case, we decided, okay, how do we really scale this up? How do we really scale up the data sets that can be made available for applications like these? So we organized the JUMP Cell Painting Consortium. It's a group of 10 pharma companies and a few academic groups led by us here at the Broad. We've created the largest soon-to-be publicly available cell painting data set. It comprises about 146,000 compound profiles, as well as genetic profiles of about a third of the genome, and this was performed across multiple sites. Through this data generation process, we aim to address several assay development questions and computational challenges in cell painting that could further accelerate its use in drug discovery. The data will be made public in a week on November 1st, so you can look out for that on that website. We think this data set will be a catalyst for machine learning scientists to collaborate on an incredibly rich set of fundamental problems, right? Learning effective representation from images, figuring out how to correct for batch effects, translating signatures across cell types, integrating data modalities, and many more applications that I heard about today. I think this list is definitely incomplete in that sense, and I hope this will spur all this and much more that we talked about. One such ongoing effort in our lab is to develop batch effect correction methods, for the most part by repurposing successful techniques from the single-cell RNA-seq community, where there's been a lot of activity in this front over the last few years, if you've been following that literature. Batch effects, especially in high-dimensional biological readouts, as many of you might know, can really blur out the signal, and they're really the bane of our existence in some sense. And our effort here is to come up with strategies that could allow both correcting for batch effects and also, more generally, integrating data across experiments so that the large data set that we've built here is really something that can be a catalyst for others to build upon. So I wanted to keep this short. I wanted to thank, that's our lab, thank our collaborators and funding sources for this work. Thank you.
Thank you both, Shantanu and Anne. We have time for questions, so please come up to the microphone. Pat?
Yeah, I guess my question relates more to classification versus regression. So you've shown a lot of great examples where you can classify cells. But what if I'm working on a drug discovery program and I want to, improve the molecule, right? So I'm not going to completely flip the phenotype, but can I, through image analysis, can I capture improvements that are sort of taking me on the road to the phenotype?
Yeah, for sure. Most of these morphological phenotypes are continuous. As a cell biologist, I can confirm. I don't know if you have anything more technical to add.
Oh yeah, I could probably just say that we sort of see, to some extent, a dose response in the phenotype, right? So if you sort of think about the phenotypic, the trajectory that the phenotypic signature takes in this high-dimensional space across dose, it's definitely, I mean, in many cases, you do see it continuous. So there is naturally just intrinsic to this high-dimensional space, there's continuity. I would also say that we don't sort of solve a regression problem there. We just sort of rely on the fact that this is a continuous space upon which the sort of the cell phenotype travels. But we do sort of directly solve a regression problem too in some cases. The cell health example, the other one, which you call Puma, where you're trying to predict assay readouts, those are entirely regression problems. I mean, in some cases, we binarize them because it's easier. But yeah,
Cool.
we do directly solve it as well.
Thank you.
Yeah.
Are there any online questions? No?
Hi,
Go ahead.
I'm Sarah Seren from Remix Therapeutics. And I was curious about the compounds you profiled. You mentioned something around 30,000 compounds. Could you give us a flavor for, does that represent like a traditional screening HDS-Sec, or is it like natural products? Or like, and also if you have any efforts to expand that chemical space to novel scaffolds.
Yeah, I love this question. So yeah, we have, up until this year, we've had 30,000 compounds to work with in the field in general, and that came originally from a project, for those who remember the MLSCN or the MLPCN consortium project through the NIH. It was a subset of compounds there. Some proportion of them were diversity-oriented synthesis as their origin, so they had some pretty, some interesting variety of structures, and then some proportion were bioactives. I don't think any natural products, but at least bioactive compounds. Now, with the JUMP consortium data, we now have 116,000 compounds, and I don't think we have any statistics yet on how those fall out. Definitely some subset are commercially available libraries, and a good chunk of them are not. So I don't know if we know anything more about where they fall in terms of structural diversity and that sort of thing.
Yeah, I don't have that yet, but I believe we'll have a lot of those statistics up in our data release preprint. So
Okay,
we should
great.
follow that up.
Any online? Nope, still? Go ahead.
Yeah, I just wanted to ask a quick question on the extendability of your screens currently to biologics or even like maybe even genetically encoded medicines as they come out. Do you think it would be pretty easy to adapt a lot of your platforms to those technologies?
Oh, absolutely.
Yeah.
Yep. So anything that you can do to cells in a dish, or even tissue samples or anything else, in theory, you can take this broad concept of image-based profiling. And cell painting as an assay has been applied to different levels of cell model systems, including kind of organoids and that sort of thing. So there's a lot of flexibility there, but fundamentally, anything that you can perturb a cell with can potentially be measured out in this way. Having good controls is critical. A lot of times it makes, instead of just saying, does my perturbation make an impact versus negative controls, it's nicer to perform an experiment where you have lots of flavors of perturbation. So, analogs that are really close but a little bit different, don't have the function or do have the function. So that's a lot nicer, I think, versus just trying to have one particular negative control, because you get a richer flavor of how the things cluster with each other. But yes, it's certainly applicable. Thank you.
Really fascinating topic. You showed Angelman syndrome, Noonan syndrome down versus normal phenotype. Assuming you're able to reverse the phenotype in cells with some chemicals, would that lead to possibly reversing the phenotype in biological organisms.
That's the hope, yes. So the hope is that any compound that we find a cell-based phenotype, we reverse it, disease is cured / treated.
Do you have an example of this where you're getting close? I'm just curious.
Yeah, so as, my lab personally doesn't, but there are some examples of this in the literature. I mean, just any experiment where there has been an image-based phenotype and then has gone on to drug discovery. But Recursion has the clinical candidates that are just entering clinical trials. So none of those have read out yet. So we don't know how it turns out. But I do want to offer a big caution. So hopefully everyone can hear the asterisk in my voice where I'm saying, ideally, the compounds that work nicely in cells in a dish, instantly translatable to humans. In reality, there's all kinds of reasons why that relationship might not hold. Not the least of which is because you have to pick a cell type to do this experiment in. And as we saw in the, I didn't show the data, but as we saw in psychiatric disorders, we can find that phenotype in fibroblasts, not the right cell type as far as you would expect, and yet we could find a phenotype, and it is showing what's wrong with that in that disorder. So I think in many cases, using the wrong cell type will actually be perfectly fine, because many of the mechanisms that underlie disorders, they might be most impactful in a particular cell type, but that doesn't mean they have no impact on other cell types that you might be testing. So sometimes it'll work, but I think that the biggest kind of way that this experiment, where that relationship will not hold is, let's say there's some kind of mechanism where what we see in cells growing in a dish is not really the symptom of the disease, it's the impact of the cell trying to overcome what's going wrong in the disease. So we might be seeing some kind of feedback loop effect that's, the cell freaking out, trying to fix the problem on its own. So if we knock that down, we might make the disease worse. And that's why I would never recommend doing an experiment in U2OS cells and then taking it right to clinical trials. In reality, you still want to go through the same sensible steps for each disorder, but at least this is a really wide, broad, systematic approach to get you to, okay, out of the 4,000 rare diseases, here's 200 of them that are tractable in this way. Now we can focus our energy on those 200 and then take them to more translatable, more physiologically relevant model systems.
Just one quick curiosity. You had thrombocytopenia. That's a clear phenotype in itself, but you had a different cell line because obviously you didn't use thrombocytes because they were absent.
I don't know.
Right?
I am sorry, I don't know which one.
It was one of the, anyway.
Yeah.
Okay.
Is there a question over here? Do you have a question?
Yeah, fantastic talk. My question might be irrelevant to the drug discovery. I think it's also a great platform to understand the disease mechanism behind. So what about if the correlation with the disease genotype actually falling into a category of features that is embeddings instead of a features that is easy to explain? How we can use that information to trying to interpret the disease mechanism?
I think I missed just the last part. Can you say the last part?
I mean, if the correlated part with the genotype actually falling into the embedding category that is extracted by the deep learning model, that is not human interpretable, how that can help us to move forward.
If I understand correctly, you're saying what if it's the case that we use a deep learning model to identify a phenotype? How do we interpret that, or what do we do with that? Do you want to? I want to give Shantanu a chance.
I think ultimately it is a hard problem to sort of come up with a human interpretable explanation for some of these things. And Andrew referred to that a little bit, saying that it is superhuman, extra human. As Neil deGrasse Tyson said, the universe is under no obligation to explain itself to us, right? So in many of those cases, you might end up with a case where you sort of see a statistically significant, consistent difference, and you're not able to necessarily translate that into biology. But as long as you're sort of able to do that reproducibly, I would say that you now have a dimension in which you can screen for the disease, screen for therapeutics for the disease, or screen for gene knockouts that might correlate with that. So it still sort of gives you a direction in the high-dimensional space to look for interpretability by matching the similarity of that to something else that you know more about, even if that specific readout itself is not meaningful. So that's the other way to think about it.
And I will say the culture has changed quite a lot. At the very beginning, interpretability was the most important thing to all the biologists we worked with. So we'd say, ooh, we found that your disease or your gene or your drug does something to cells. And their first question is, what does it do to the cells? And they really wanted to understand what the mechanism is. But as we've gone on and had some more machine learning intensive applications where we just say, oh, well, there's some information in this image that predicts the readout in this other assay. We don't really care what it is. As long as we predict this outcome in this assay we care about, then we're good to go. So interpretability has become a little less important as people have adopted to the idea of using this as kind of a quantitative data source.
Hi, both of you, great talk. I just had a very layman question that came in my mind. That is, I would imagine that this will be dependent on a distinctive morphological feature. Have you experienced a case where the cell does not display any morphological features?
Oh, well, there's certainly, roughly 50% of the time there's no impact on the cell that's detectable to us at all. But then there's often, if you're sort of asking what proportion, when there is a detectable change, what proportion of the time is it detectable to our eyes versus the computer? I'm not sure if I have a good percentage of that. Biologists are very good at making up stories. So a lot of times we'll look at images and say, okay, the computer tells me that these are different, and now I'm staring at them and like, yeah, I see a difference. So it's a little harder to say, but I guess if I had to guess, it's probably more than half the time it's not so discernible to the eye. And partly that's just because not because we can't see the thing, but more because of just the statistics. It's two heterogeneous populations that differ just slightly. It's statistically detectable, but it's not the thing that our human brain is so great at capturing.
And would you say that the morphological features become more distinctive for rare diseases versus common diseases, or any from your experience?
I love that question, and I don't know the answer to it. But one could imagine that rare diseases might be more detectable because they tend to have more extreme phenotypes in the humans. But it's hard to say. Most common diseases have, of course, a collection of variants.
Perhaps the only other thing I can add is that the operator variance matters a lot. I think things that Anne can see, there's no way I can see it. Just looking at it, said I have no idea. So I rely much more in that sense on the statistics. But the other aspect of it was also that, to some extent, you don't want to be able to, figure it out, get some sort of sanity check that you can see the phenotype. But, as you go more and more multidimensional, multi-channel, now you can do screens with, 20 markers, right? At that point, when you're looking at a 20-channel image, then you gotta sort of rely much more on the statistics as you go. No doubt human expertise should be baked into trying to go from the image to the biology, and that's important. But at some point, that does break down.
All right, I think that's all the time we have for questions, and thank you so much, Anne and Shantanu.
Thank you.